Somewhere in a lab, a geneticist stared at a spreadsheet and stopped breathing for a second. Not because a machine had broken. Not because a sample had been contaminated. Because the numbers in front of her were saying something that shouldn't be possible. An entire population, thousands of people, and almost none of them carried the blood types the rest of the world considers normal. For a moment she assumed it was an error. It wasn't. And once you understand why, you will never look at your own blood type the same way again. Most of us learned our blood type the way we learned our shoe size. A nurse pricks a finger, a card gets stamped with a letter and a symbol, and that's the end of it. A, B, AB, O, positive or negative. It feels like trivia. Something you might need once in your life if you're ever in an accident and a stranger's blood has to match yours. Nobody tells you that those letters are actually a kind of fossil. Not a fossil made of stone, but one made of biology, carried inside every one of us, quietly recording where our ancestors lived, what diseases nearly wiped them out, and which ancient roads their bloodlines traveled long before anyone wrote anything down. Blood type isn't random. It's a map. And once scientists started reading that map properly, comparing it across populations instead of just within them, the picture that emerged was far stranger and far more revealing than anyone expected. For most of the 20th century, the working assumption in medicine was straightforward. Blood type is essentially a coin flip. Every population should have roughly the same mix of AB, and O, because there's no obvious reason nature would favor one letter over another in one place, and not somewhere else. That assumption held up fine as long as nobody looked too closely. But geneticists and population biologists have spent decades quietly building databases of blood type frequencies from every corner of the planet. Indigenous communities in the Americas, herding populations on the Central Asian steppe, villages across West Africa, isolated groups tucked into European mountain ranges. And when you actually lay those numbers side by side instead of averaging them into one global blur, the coin flip story falls apart almost immediately. Start with the type that most people think of as the boring, default option, type O. It's called the universal donor because, in an emergency, it can be given to almost anyone regardless of their own type. Globally, it's also the single most common blood type, so on paper it looks unremarkable, just the most popular option in a large, evenly mixed crowd. But break the global average down population by population, and something happens that shouldn't happen if this were truly random. In several indigenous populations of the Americas, groups like the Navajo in the American Southwest, various Amazonian communities, and Andean populations in South America, the frequency of type O doesn't just lead the pack. It approaches, and in some documented groups, essentially reaches near total dominance. Researchers studying certain isolated indigenous populations have reported vanishingly small numbers, sometimes none at all, of confirmed type A or type B individuals. If blood type were a genuine coin flip, this is the equivalent of flipping a coin 50,000 times and getting heads every single time. That doesn't happen by chance. Something forced it. The leading explanation is what geneticists call a founder effect layered on top of a bottleneck. When humans first crossed into the Americas from Asia, likely across the Bering Land Bridge, or along the coastline during the last ice age, they came in small groups. Small founding populations carry only a fraction of the genetic variation of their larger parent population, purely by chance. Imagine reaching into a jar of mixed marbles and pulling out a handful. You probably won't get a perfectly proportional sample of every color. If the founding groups that populated the Americas happen to carry mostly the O-type gene variant, and if those small populations then remained relatively isolated for thousands of years afterward, that narrow starting point would simply persist and even intensify across the generations that followed. Some researchers add a second layer to this explanation, that disease outbreaks in the deep past may have disproportionately affected people with A or B markers, tightening the population's genetic profile even further over time. Neither explanation is proven beyond doubt, and most scientists in the field are careful to say so. But the pattern itself, the wall of type O, is not in dispute. It's one of the most striking blood type findings in the world, and it sat there in plain sight for decades before it collected the attention it deserved. If type O could draw a wall that sharp, the next question was whether the supposedly random letters A and B might be hiding maps of their own. And when researchers looked, they found gradients, smooth, consistent slopes rather than scattered noise. Among populations of European descent, type A frequency tends to sit somewhere in the low to mid-40s as a percentage. Move east across Asia, and that number steadily drops, often into the high 20s. Continue into parts of sub-Saharan Africa, and it falls further still. This isn't random static bouncing around an average. It's a slope, a gradient that declines steadily the farther you travel from a particular region, as though the gene had an origin point somewhere and thinned out the farther it spread, the genetic equivalent of ripples fading as they move away from where a stone hit the water. Type B tells an even more vivid story because its geography lines up almost perfectly with a very specific and very famous chapter of human history. In Western Europe, Type B is genuinely rare, often below 10% of the population. But travel east into the vast grassland corridor that stretches from Ukraine through Kazakhstan and into Mongolia, and the numbers climb sharply. In some Central Asian populations, Type B frequency reaches into the 30s, 3 or 4 times what you'd find in Western Europe. That corridor isn't just any strip of land. It's the historic Eurasian steppe, the great highway that carried migrating and conquering peoples back and forth across the continent for thousands of years, most famously during the 13th century Mongol expansion, when a relatively small population out of the eastern steppe built the largest contiguous land empire in history in only a few generations. Blood doesn't get rewritten the way history books sometimes are. When a population moves and mixes with the people it encounters, it leaves a genetic signature behind that can still be measured centuries later. Several population geneticists have pointed out that the modern distribution of Type B frequencies across Eurasia roughly tracks the historical routes of steppe migrations, with the heaviest concentrations closer to the core migration corridors and a gradual tapering the farther you move from them. It's a reasonable and widely discussed hypothesis, though, like most deep history genetics, it's built from patterns and probabilities rather than a single smoking gun piece of evidence, which is exactly why scientists studying it tend to speak in terms of consistent with rather than proof of. Then there's the Rh factor, the plus or minus sign most people barely register on their blood type card. Rh-negative blood is uncommon almost everywhere. In European-descended populations, it shows up in roughly 15% of people. In sub-Saharan Africa, that number is lower, and in East Asian and indigenous American populations, it often drops below 1%, making it close to a genuine rarity across most of the human species. Which makes one population's numbers stand out immediately. The Basque people, an ethnic group living in the Western Pyrenees along the border of Spain and France, whose language is famously unrelated to any other known language family on Earth. Multiple population studies have found Rh-negative frequencies among Basque populations sitting well above the European average, with some samples reporting figures in the region of 25 to 30% or more, a striking outlier by any standard. The explanation that's gained the most traction among researchers ties the Basque anomaly to the deep history of Europe itself. Roughly 8,000 years ago, farming populations began spreading into Europe from the direction of Anatolia, part of a slow agricultural migration that eventually mixed with, and in most regions substantially replaced, the hunter-gatherer populations who had lived there for tens of thousands of years before them. Across most of the continent, that older hunter-gatherer genetic signature was diluted and blended into near invisibility by the incoming farming populations. But the Pyrenees, protected by geography, and the Basque language, which never merged into the wave of newcomers' tongues, may have preserved a genetic profile that elsewhere got smoothed away. If that's correct, the elevated Rh-negative frequency isn't a fluke or a lab error. It's an echo, a genetic remnant of a Europe that existed before farming ever arrived. It's one of the more compelling examples of how population genetics and linguistics, two fields that rarely cross paths, can independently point toward the same conclusion about a group's deep isolation. The most medically important thread in all of this, though, involves a blood group most people have genuinely never heard of, the Duffy antigen system. The Duffy antigen is a protein sitting on the surface of red blood cells. Most humans carry it without any awareness of its existence because it doesn't show up on a standard blood type test the way A, B, or Rh does. But its distribution across human populations is one of the most extreme and best understood patterns in all of human genetics. There's a variant known as Dufenol, in which that antigen is essentially absent from red blood cells. In populations with Deep West and Central African ancestry, which includes a large share of African American populations today, the Dufenol variant is extremely common, showing up in the large majority of individuals in many studied groups. In populations of European or East Asian descent, the same variant is exceedingly rare, often under 1%. That's not a mild statistical tilt. It's close to a binary split between different parts of the world. Unlike the type O wall, which still doesn't have a fully settled explanation, the Duffy pattern has one that scientists consider essentially proven: malaria. Specifically, a form of the disease caused by the parasite Plasmodium vivax, which needs the Duffy antigen as a kind of docking receptor to force its way into a red blood cell. No Duffy antigen means no docking point, which means no entry, which means no infection, at least from that particular strain. In regions of West and Central Africa where Vivax malaria was a relentless killer for thousands of years, carrying the Dufenol variant wasn't a neutral genetic quirk. It was the difference between surviving to have children and not. Over enough generations, that kind of unforgiving selective pressure reshaped the genetic profile of entire regions. It's one of the clearest, best documented examples of natural selection acting on the human genome within a relatively short evolutionary timeframe, and it's been studied and confirmed by multiple independent research groups over the past several decades. Here's where the story stops being ancient history and becomes something that matters in a hospital today. When new drugs are developed and tested, their safe and effective doses are established through clinical trials, carefully measured studies where researchers observe how real human bodies process a given compound. For much of the history of modern pharmaceutical research, the people enrolled in those trials came disproportionately from populations of European descent, in whom the Dufenol variant is nearly non-existent. Some medical researchers have pointed out that certain drugs affecting white blood cell counts, for example, can look artificially abnormal against dosing and reference ranges built primarily from European ancestry populations, when what's actually being measured is a normal variation tied to Duffy status that's common in populations of African descent. This has real clinical consequences. A benign, harmless variation in some people's blood counts has, in specific documented cases, been misread using reference ranges that weren't built with a full range of human biology in mind. It's not a story about malice. It's a story about how medical standards, like almost everything else, get built from whoever happens to be in the room when the data is collected, and the fact that we're still catching up to that reality is worth sitting with. There's one more thread worth mentioning honestly, because pulling on it carelessly is how good science turns into bad headlines. The question of who ancient populations were related to, and how that connects to people living today. Advances in the last few decades have made it possible to recover blood type and broader genetic information from ancient remains, bone, teeth, and in some cases preserve soft tissue, allowing scientists to compare the blood of the living against samples thousands of years old. This kind of ancient DNA work on populations like those of ancient Egypt has produced genuinely fascinating and genuinely contested results. Different studies, using different methods and different ancient samples, have found different things, and serious researchers in the field disagree with each other about how to interpret ancestry signals from a region that sat at the crossroads of Africa, the Near East, and the Mediterranean for thousands of years. The honest, responsible summary is this: ancient Egypt was genetically diverse and shaped by migration and trade from multiple directions over thousands of years. The scientific picture is still being actively refined as more ancient samples are analyzed, and any confident, simple claim about a single true origin for an ancient civilization is almost certainly oversimplifying real, ongoing scientific debate. That nuance isn't a disappointing footnote. It's the actual state of the science, and it deserves more respect than a dramatic headline. What ties all of this together isn't a single discovery. It's a shift in perspective. For a long time, blood type was treated purely as a personal, almost trivial fact, useful for a transfusion, and nothing else. But zoom out from the individual to the population, and from a single generation to tens of thousands of years, and the letters stop looking arbitrary. They start looking like layers of sediment, each one laid down by a different chapter of human history, a migration into an empty continent, a disease that reshaped an entire region's genetics, an empire that rode out of the steppe and left a biological signature 8 centuries deep, a mountain range that sheltered a language and a bloodline from being absorbed into the wave around it. None of this is about ranking populations or reviving old, discredited ideas about race as some kind of rigid biological category. In fact, it's almost the opposite. The deeper you go into this data, the clearer it becomes that human populations blend, move, and share ancestry in complicated, overlapping ways that don't map neatly onto old categories at all. What the data actually shows is a single, continuous human story, one where every population's blood carries traces of survival, movement, and adaptation stretching back far further than any written record. So, here's the question worth sitting with. The next time you see your own blood type on a card or a lab report, what does it actually mean to you? Is it just a compatibility code for an emergency room? Or is it something closer to a fragment of a much longer story, one written in malaria survival, in ancient migrations, in mountain ranges that kept a bloodline intact for thousands of years? What's your blood type, and does knowing even a little of what it might represent change how you think about it? Drop your type in the comments, and if this kind of deep history science is something you want more of, you know exactly what to do next.